Dear students, we have now come to the second half of the final unit. Next, the teacher will introduce apoptosis and autophagy. So, what's the difference between apoptosis and autophagy? Both apoptosis and autophagy naturally occur inside our cells. They are processes of self-degradation. In fact, they help maintain a balance in the life cycle of our cells. Regarding apoptosis, I've already introduced a lot to you in the first half. Actually, it is a process of cell suicide. It follows a certain program and is regulated by certain genes. It also plays a very important role in cancer. So what about autophagy? Autophagy is a process of self-degradation in which the cell balances the energy supply during development. Autophagy degrades unnecessary or dysfunctional cellular components, which helps the cell survive under stressful conditions. It basically engulfs some of its own organelles and stays alive. This is what we call cellular autophagy. Cellular autophagy. So what role does it actually play in cancer? Autophagy. In the suppression of early tumors, it can inhibit the formation of early tumors, but once the tumor has formed, autophagy may actually promote tumor development and maintenance. So students, you can think about this. Have you ever heard someone say, I have cancer, so I'll just stop eating and starve the cancer cells to death? This is a very wrong idea. Before you even starve the cancer cells, your own immunity might already decline. Why is that? Because inside cancer cells, when they are in a starved state, they can use autophagy to engulf their own organelles and still survive happily. So never think, I'll starve myself to kill the cancer cells. It's impossible. Now, chemotherapy and radiotherapy can induce autophagy, which has a protective effect. If you inhibit autophagy, it can increase the sensitivity of tumor cells to treatment. Later, the teacher will explain the role of autophagy in cancer therapy. Since cancer cells experience nutrient deprivation, growth factor exhaustion, metabolic stress, hypoxia, or are exposed to anti-cancer drugs, targeted therapy, or radiation, autophagy will be induced. The more you try to kill me, the stronger I survive. This is what makes cancer cells so difficult. Usually, autophagy is connected to some drug resistance. It can act as a cancer-promoting factor, affecting the final outcome of cancer treatment. Now, the teacher will first explain what drug resistance is. Drug resistance, simply put, is when you take a drug, at first it kills, say, 10,000 cancer cells, the second time, third time, fourth time, fifth time. When taking these drugs continuously, gradually, some of the cancer cells will start to accumulate gene mutations. The same dose may no longer kill all 10,000 cancer cells because these cancer cells have also developed mutations, genetic mutations. It's possible that the same dose may now only kill 1,000 cancer cells because these cancer cells have developed drug resistance. Autophagy is actually closely related to drug resistance. Scientists have found that during treatment, cancer cells induce autophagy to survive. Given this, inhibiting autophagy in cells can of course enhance the effectiveness of anti-cancer drugs. Later on, in different types of cancers, some trials confirm this. For example, quinine compounds, chloroquine, and hydroxychloroquine. Traditionally, these are used to treat malaria or rheumatoid arthritis. Later, it was confirmed that they have the function of inhibiting autophagy. So when a cancer cell needs autophagy to survive, you can use chloroquine or hydroxychloroquine in combination. At this point, this can induce the cancer cell to undergo apoptosis. So in a cancer cell, autophagy actually acts as a mechanism that helps the cancer cell survive. How does autophagy occur? Earlier, the teacher mentioned apoptosis. Apoptosis can occur through exogenous and endogenous pathways. The exogenous pathway is through receptors and activators, which then induce a cascade activation of caspases. The endogenous pathway involves molecules that are pro-survival or pro-apoptotic, which can induce the release of cytochrome C, then activate caspase 9, and then caspase 3. Autophagy also occurs through a series of mechanisms. It can be divided into three main types: macroautophagy, microautophagy, and chaperone-mediated autophagy. Through autophagy, damaged organelles, misfolded proteins, and other macromolecules are ultimately degraded via lysosomes. Thus, autophagy plays an important role in cell growth, development, and homeostasis. It is also involved in angiogenesis, osteogenic differentiation, and neurogenesis. The main function of macroautophagy is to clear damaged cellular organelles and unused proteins. First, the phagocytic cell engulfs the material to be degraded. Then around the damaged organelles, a structure called the autophagosome is formed. This autophagosome. Then, through the cell's cytoplasm, the autophagosome reaches the lysosome. These two organelles fuse together, and inside the lysosome, the contents of the autophagosome are degraded by acidic lysosomal hydrolases. This is what we call macroautophagy. So, what is microautophagy? It occurs through invagination or folding of the lysosomal membrane or the cell protruding outward, which directly engulfs the material to be degraded into the lysosome. This is the main difference between macroautophagy and microautophagy. Next, let's look at the role of apoptosis and autophagy in the development of anti-cancer drugs. For apoptosis, most anti-cancer drugs usually work by inducing cancer cells to undergo apoptosis, thus killing them. However, some cancer cells, as the teacher mentioned earlier, develop gene mutations that lead to drug resistance. So understanding how apoptosis occurs helps scientists develop anti-cancer drugs. Autophagy, on the other hand, is usually an important early mechanism in cancer. It plays a dual role. As I mentioned before, once the tumor has already developed, if the cancer cell is treated with chemotherapy or radiation, or is in a starved state, it can use autophagy to survive. So autophagy can be divided into two stages. One is early tumor suppression, and the other, once cancer has developed, it may play a role in drug resistance. Next, the teacher will introduce a study published in a scientific journal from Tokyo Medical and Dental University. They designed a so-called antibody supermolecular complex, which targets cancer cells via the autophagy death pathway. This new type of antibody drug conjugate, abbreviated ADC, is a complex of antibody and drug. It can kill cancer cells through autophagy and has a longer duration of action, which helps it be absorbed by tumors. So, as mentioned earlier, understanding autophagy can help us develop anti-cancer drugs. Now, what role does apoptosis play in drug resistance? When the apoptotic pathway fails, for example in chemotherapy or targeted therapy, the treatment is supposed to kill cancer cells through apoptosis. But sometimes the human body produces anti-apoptotic factors. Earlier, the teacher introduced some pro-survival molecules, BCL2, BCL-XL, and the so-called IAPs. IAPs are caspase inhibitors. These factors can control the mitochondria to release cytochrome C, inhibit apoptosis, and lead to drug resistance. In addition, in the teacher's research, it can also be observed that when our DNA repair system is overactivated and when tumor cells' DNA repair system mutates, it becomes hyperactive, which can cause these drugs to fail to kill cancer cells and result in drug resistance. So drug resistance is not only caused by failure of the apoptotic pathway, but also by overexpression of DNA repair. To give an example of DNA repair overexpression, students can think about this. Some chemotherapy drugs mainly aim to kill cancer cells. These chemotherapy drugs affect the DNA. Some cause DNA damage, and DNA damage triggers DNA repair. It's like if I get injured today, I would need a doctor to stitch my wounds or perform surgery. Now think about this. Inside a cancer cell, would you want to give it a top-notch doctor or a mediocre doctor? Of course, you would give it a mediocre doctor, ideally an incompetent one. An incompetent doctor, after the cancer cell is injured, won't really heal the cancer cell, but cancer cells are very clever. They don't choose incompetent doctors. Instead, they recruit many doctors to repair their damaged parts. So to survive, the cancer cell is really smart. It disables the apoptotic pathway, and it activates its DNA repair system, allowing it to resist drugs. Understanding these mechanisms is necessary to combat drug resistance. In addition, there are tumor stem cells. Tumor stem cells are the most primitive cells that can develop into a tumor. They express drug resistance proteins like P-glycoprotein. These stem cells are resistant to chemotherapy drugs, so even if most cancer cells are killed, tumor stem cells still express drug resistance proteins, allowing them to regrow, which leads to tumor recurrence or metastasis. So, students, as mentioned in previous units, when receiving treatment, you may maintain a period where the prognosis is good, meaning the tumor does not regrow. But this does not guarantee that all cancer cells are killed. Tumor stem cells may still exist. When your immune system weakens or your body condition worsens, these cancer stem cells can regrow, leading to tumor recurrence. Now, the tumor microenvironment is often hypoxic. The tumor microenvironment is usually hypoxic. This hypoxic state induces a molecule called HIF. HIF has many effects, including changes in metabolic functions, overexpression of multiple drug resistance proteins, and inhibition of apoptosis. So usually, the hypoxia in a cancer cell's environment is highly related to cancer progression and even distant metastasis. Next, the teacher will give a summary of the final unit. Apoptosis is the main mechanism to eliminate cancer cells. Through apoptosis, we can reduce tumor size and enhance the effectiveness of treatment. So whether it's chemotherapy, radiation, or targeted therapy, the goal is to make the cancer cell undergo programmed cell death. Earlier, the teacher introduced autophagy and apoptosis and explained how they are different from necrosis. In the second half of this unit, the teacher introduced autophagy. Autophagy plays a double-edged role in tumors. In the early stages of cancer, it can prevent tumor formation and limit the accumulation of damaged materials. But once the tumor progresses to the late stage or is under environmental stress, autophagy may help the tumor survive. So never think that starving the body will kill cancer cells. On the contrary, it can provide energy to cancer cells through autophagy, allowing them to survive, invade, and metastasize. Our unit series is now complete. From Unit 1 to this final unit on apoptosis, we hope students now have a clear understanding of how cancer develops, and we hope that Cancer Unlocked has truly demystified cancer for everyone. Thank you, everyone.